In this video, we are going to explore the topic of microscopy, which simply refers to the use of microscopes to observe very small objects that cannot be seen clearly with the naked eye. We will focus on how light microscopes work and clarify several important terms including object, image, magnification, and resolution. To begin with, let us understand the main parts of a typical light microscope. At the very bottom is the base, which supports the entire structure, and connected to it is the arm, which joins the base to the upper section of the microscope. For lighting, some microscopes include a lamp, but others, like the one shown here, use a mirror to reflect light from the room upwards through the specimen. Directly above the mirror is the stage, which is the flat platform where the microscope slide is placed. Moving to the upper part of the microscope, you will find several lenses. Typically, there are 3 objective lenses of varying magnifications, such as 10 times, 20 times, and 50 times. These can be rotated and selected depending on how much you want to magnify your specimen. In addition to the objective lenses, there is an eyepiece lens with a fixed magnification located at the top, which is where you place your eye to look into the microscope. Other components include the body tube that connects the eyepiece to the objective lenses and 2 focusing knobs known as the coarse focus and the fine focus, which help in sharpening the image of the specimen being viewed. Before diving deeper, we need to explain the terms object and image. In the context of microscopy, the word object refers to the actual specimen or physical material on the slide that we are observing. For example, if we are looking at onion cells on a slide, those cells are the object. In contrast, the term image refers to the enlarged visual representation of the object that we see when we look through the microscope. So, when you look into the eyepiece and see the enlarged version of the onion cells, what you are seeing is the image. It is important not to confuse the object, which is the physical sample, with the image, which is the enlarged view. Now, let us see how a light microscope functions. Light from the environment hits the mirror and gets reflected upward through the specimen that is placed on the stage. In this case, it would pass through our onion cells. After that, the light continues through one of the objective lenses, then through the eyepiece lens, and finally into our eyes. The main idea here is that the lenses bend or spread out the light rays, allowing us to see an image that is much larger than the actual specimen. This brings us to the concept of magnification. Magnification is defined as how many times bigger the image is compared to the actual object. For example, if the image looks 1,000 times bigger than the object, then we say the magnification is times 1,000. There is also a basic formula for calculating magnification. It states that magnification equals image size divided by object size. We will cover how to use this formula with actual numbers in another lesson. Another important term in microscopy is resolution. Resolution is defined as the shortest distance between 2 points on an object that can still be seen as separate points in the image. While this definition sounds complex, it simply means how close 2 features on an object can be while still appearing distinct from one another. In simpler terms, it measures the clarity or detail of the image. For instance, if we look at 2 microscope images of the same onion cells, both magnified 100 times, they may appear the same size, but one may look sharper and clearer than the other. The clearer image has a higher resolution, while the blurrier one has a lower resolution. This means it cannot show as much fine detail. So, always remember that a higher resolution allows for more visible detail and reduces blurriness. To summarize, a light microscope lets us observe objects that are otherwise too small to see with the unaided eye. It uses light and lenses to magnify and produce a clear image of the specimen, allowing us to study features at a cellular level. Understanding the concepts of object, image, magnification, and resolution is key to interpreting what you see under the microscope accurately. That covers everything for this video on microscopy. If you want to test your knowledge or practice questions related to this topic or others in science, simply click on the link provided in the top right corner or check the pinned comment below. Hopefully, we'll see you again in the next session.